So I'm going to go skip from this title slide, tell you a little bit about myself and my journey through science, and then how I happened upon being a part of this really exciting project that I get to tell you about. So my dad was born in Dominica, a small island in the Caribbean. My mom was born in Jamaica. You guys are probably familiar with that island. And they both immigrated here to the United States. And then my dad joined the military, and they met here in Florida. My childhood was basically moving around from place to place because my father was in the Navy. And I spent majority of my childhood, I would say, growing up in Pensacola, Florida, near the Navy base there. In school, I always loved science so much. So I ended up enrolling in a undergrad program at the Southern Adventist University in Tennessee. So it's a small private institution. I had a couple athletic scholarships in order to go to the school, and I just completely fell in love with science. And my second year in undergrad, I was able to do some undergrad research and found that I really, really love research, but I had no idea what I wanted to do moving forward. So through some happenstance that I really don't have time to get into, I decided that I wanted to go to graduate school to study type 2 diabetes, which is a disease that's really prevalent in my family. My mom has diabetes, my grandma, a couple of my aunts. I'm pre-diabetic. So it's just kind of everywhere, and I really have a passion for understanding metabolic disease and type 2 diabetes in general. So I took a big move across the country to Loma Linda University, and I did a graduate program there. So while in graduate school, my love of science never diminished. It didn't change, but I did get a little disillusioned with, I guess, my path through science. It seemed like, at least from the perspectives of people I met at conferences, from the PIs who were there and running labs, that first of all, it's very hard and time consuming in order to have your own laboratory and do your own research, get your own grants. It's going to be especially difficult if you're a woman who wants to have a family or who wants to have a life period, and they expect you to put in hours and hours and hours into lab, and there's no emphasis on work-life balance. And I found this really disheartening because growing up, I always imagined having a family one day and still being able to run my own lab. So while in graduate school, I actually met a woman who really, really inspired me. And I got to do my postdoc with her at Boston University. So I'm going to talk a little bit more about this particular PI in the next slide, and why she really made a big impact on me in the project that I worked on while in her laboratory. But following that postdoc at Boston University, I was able to do a postdoc here at UCSD in Mark Lawson's lab and co-mentored by Pamela Mellon, where they've been super supportive and have basically helped me blossom into what I say is a really strong, independent scientist with a lot of confidence. And I'm really pleased to announce I'll be able to start my own lab at UC Irvine next summer. So my first postdoc was with Dr. Barbara Nikolajczyk at Boston University. And she, to me, was an example of a woman who did great science, well-known, networked, and who also had a beautiful family. So she has 2 sons. She would take vacations. She was very supportive of other women in the lab who had families, and was expecting that we would get our work done and we could meet and talk one-on-one, things like that. But then also that when you have personal priorities, that those priorities were important, and that science isn't all of life, that there's multiple dimensions to us as people. So as I began working with her, she did something that I thought was really, really important, at least for me, is that she spoke to me as a colleague. She respected my ideas. She looked to me for my expertise, and then spoke with me one-on-one, back and forth, to a point where she's like, okay, Dequina, this is your project, you take the lead, and I will help you get wherever it is that you need to go. So throughout the project that I'm about to show you, we actually ended up putting together a really big network, or a huge collaboration among a lot of different scientists. And I felt it was truly, truly amazing. So here's photos of majority of the authors from this paper that we published just last year. And the authors, as you can see, are very diverse. They span ethnicity, religion. We actually have sexual orientation differences, changes, gender, different backgrounds in scientific disciplines. We have bioinformatics, we have MDs, we have PhDs, we have grad students, we have master students. And just working with this group basically gave me a key thing that was so important for me in science that I didn't have during graduate school, was that working with such a big group that was so diverse, that all respected each other, gave me a sense of belonging in science. I felt like, as a Black woman, I belonged in science, my ideas were valued, and it gave me confidence to move forward and move this project forward. So with that said, this project is based off of human samples from people with type 2 diabetes. And as I mentioned, many people in my family have type 2 diabetes, which is why I wanted to be in a laboratory that was at the forefront of understanding what is the etiology of the disease. So there are several risk factors for type 2 diabetes. One is a lack of exercise, which promotes obesity. Also your diet, if you have poor diet and high fats, your race and family history. So for me, African Americans have a high risk for type 2 diabetes, so Native Americans, Hispanic Americans. Also age, the older you are, the more likely it is that you'll develop type 2 diabetes. And then having just gestational diabetes is also a risk factor for developing type 2 diabetes. And what ties a lot of these risk factors together is that we know that there's chronic inflammation that can actually lead to increased amounts of insulin resistance in type 2 diabetes. So the field has basically known this for about a decade. The inflammation is central to the development of type 2 diabetes. However, we've made very, very little progress in targeting and treating inflammation in type 2 diabetes. And why is that? So we believe that part of the reasons are historical. So early on, the first molecules discovered to be associated with inflammation in type 2 diabetes were certain cytokines. And cytokines are molecules that are secreted by immune cells for cell-to-cell communication. What I have here on the screen are 3 different cytokines that were historically the first to be related to insulin resistance in type 2 diabetes. We have tumor necrosis factor alpha, or TNF-alpha, interleukin-6, or IL-6, and interleukin-1 beta, or IL-1 beta. And what has happened is that in the clinic, they have tried to individually target these cytokines with little impact on insulin sensitivity and impacts on management of type 2 diabetes. So it kind of seems like it's a failure. However, when they tried to treat mice, they found that, okay, we can basically ablate type 2 diabetes in mouse models. However, we know that mice aren't people, even though many times mice can be a really, really great model for studying human disease. So what is this difference between mouse and humans that is basically holding us back in targeting inflammation in type 2 diabetes? For the mouse model, a diet-induced obese mouse is the gold standard for assessing type 2 diabetes. But what we do know about this mouse model, as opposed to human type 2 diabetes, is that the inflammatory process is innately different. So within the immune system, there are many different types of cells, and one subset of these cells are called T cells. And T cells also have many different types of functions that they can do depending on their phenotype. So I'm not going to go over all the different subsets and T cell biology that could possibly exist. We're going to just touch on 2 which are important for this talk today. So one subset of immune cells are TH1 cells. And TH1 cells secrete cytokines such as IL-2, interferon gamma, and TNF-alpha. So these cytokines that a particular subset of T cells secretes gives it a particular cytokine profile. So we call this the TH1 cytokine profile. TH1 cells are dominant in the diet-induced obese mouse model. They're responsible for cell-mediated immunity, ultra-insular pathogens, and then really in particular, they have acute inflammatory responses, or associated with acute inflammatory responses. In human type 2 diabetes, however, instead of a TH1 type of inflammation, we see a Th17 type inflammation. And Th17 T cells secrete cytokines such as IL-17, IL-21, and IL-22. So these are very distinct cytokines that will let us know that a Th17 type of inflammation is occurring. And what I want to point out about Th17 cells is that they're very much associated with autoimmunity and chronic inflammatory responses, much like of what we see in human type 2 diabetes, of course. So knowing this, that the immune cells or the immune or inflammation between mouse and human diabetes is different, we really wanted to understand why, what could be driving these differences. So one thing that we're getting to know in the field of what we call immune metabolism, which is looking at the metabolism of immune cells, is that the metabolic program for each T cell type is important for T cell function. So for example, here I have an effector T cell, and in the field, we kind of accept that glycolysis is important for effector T cell function. On the other hand, we also have regulatory T cells, which are anti-inflammatory, and we know that fatty acid oxidation is important for supporting these types of T cells. So that made us wonder whether or not cellular metabolism could be responsible for Th17 inflammation that we see in human type 2 diabetes. What we wanted to look at was the differences that we see in human serum during type 2 diabetes, such as increased glucose, increased free fatty acids, and increased branched chain amino acids. And we wondered if these changes in fuel source could actually impact T cell function. So our hypothesis was that the type 2 diabetic milieu that is different within a person with this disease can support pro-inflammatory T cell metabolism and downstream function. And by function, we mean their cytokine profile, and that they would secrete Th17 cytokines. So through serious experiments I definitely won't have time to talk about today, we're able to rule out branched chain amino acids as supportive for T cell inflammation in type 2 diabetes. So our next question was to look at overall metabolism within these immune cells from subjects with type 2 diabetes. So we used a technique called extracellular flux analysis. And this analysis can measure two major energy pathways of the cell. The first being aerobic respiration, which we measure by the oxygen consumption rate, because for fatty acid oxidation to occur, you need to use oxygen within the cell. The second is glycolysis, which we can measure by the extracellular acidification rate. So once glucose is broken down within the cell to pyruvate and then converted to lactate, we have the release of protons, which then acidify the media, and thereby we can measure the ECAR. So using this technique, we can simultaneously look at the oxygen consumption rate, or OCR, and the extracellular acidification rate, or ECAR. So what experiment we did was to take immune cells from subjects with and without type 2 diabetes. We then stimulated them with a T cell directed stimuli to activate only T cells within this group of mixed population of cells. Then we performed a mitochondrial stress test. So here to the left, I have a mitochondrial stress test. You don't really need to know the protocol for running this test to understand that if you look at these two lines, the blue for non-diabetic and the red for type 2 diabetic, there's absolutely no difference between these curves for oxygen consumption rate. However, if we look at the extracellular acidification rate, a representative of glycolysis, we see that the cells from subjects with type 2 diabetes have an increased level of glycolysis, therefore telling us that stimulated T cells from subjects with type 2 diabetes are highly glycolytic, more so than non-diabetic cells. So that gave us a good inkling that, okay, glucose is likely to drive type 2 diabetic inflammation. So we wanted to directly test this and know whether or not glucose was responsible. So to test whether or not glucose was responsible, we went ahead again and got immune cells from subjects with and without type 2 diabetes, and then stimulated them with T cell directed stimuli. But this time, we removed the glucose from the media. During this glucose starvation, after glucose starvation and the stimulation, we took this supernatant from these immune cells, and then we analyzed the supernatant for secreted cytokines, 25 different ones at the same time, and then performed a supervised clustering analysis. This clustering analysis is called partial least squares discriminant analysis, or PLSDA for short. I'm pretty sure several of you have heard of principal component analysis. PLSDA is a similar way to cluster multidimensional data, except that with PLSDA, this method is actually supervised. So our algorithm knows which data points belong in what group as it analyzes. And the point of this analysis is to figure out what variables are most important for distinguishing between the two groups that we have in our analysis. So for example, one data point would basically represent the 25 different cytokines for each independent sample that we have. And then what we would do through this algorithm is take all these data points that are here in the cloud and rotate them in 3D space until we find a dimensional axis that allows us the most or maximum separation between the two groups that we have previously defined. So in our case, that's non-diabetic and type 2 diabetic. So what we would expect if glucose is important for type 2 diabetes inflammation, if we've removed glucose from the equation, and we should no longer be able to distinguish between disease and non-disease. So when we actually ran this experiment, we did not see that at all. What we found is that if you glucose starve these cells, we can still very clearly have a disease-specific separation between our cohorts. So non-diabetic here on the left, type 2 diabetic here on the right. And what this is telling us is that glucose does not support type 2 diabetes associated cytokine secretion. And as I mentioned, if it was important, then we'd no longer be able to tell these two groups apart. So essentially what that did is knock out glucose as a potential fuel source that is driving or supporting T-cell mediated inflammation in human type 2 diabetes. And that leaves us with fatty acids as a fuel source. So we're going to take a little bit of time and going to some important mechanisms of fatty acid transport for fatty acids to be used as fuel within the cell. So fatty acids can't just get into the cell and then be used for fuel. They need to be actively transported into the mitochondria to be used. So after they are broken down into fatty acyl-CoA within the cytoplasm, fatty acyl-CoA must then undergo a change by the enzyme carnitine palmitoyl transferase 1, or CPT1. And CPT1 is the rate-limiting enzyme for fatty acid oxidation. CPT1 converts fatty acyl-CoA into fatty acyl-carnitine. So this fatty acyl-carnitine that's within this, between the membranes of the mitochondria, then needs to be shunted into the inner mitochondrial matrix. And this occurs through carnitine acyl-carnitine translocase, or CACT. This translocase allows the transport of fatty acyl-carnitine into the inner mitochondrial membrane. Thereby, this molecule can then undergo beta oxidation. So what we wanted to do was see if any of these important proteins for fatty acid transport were changed in subjects with type 2 diabetes. Cells from subjects with without type 2 diabetes by Western blot, and we found that we had an increase in CPT1, that rate-limiting enzyme for fatty acid transport, as you can see here illustrated to the right. Not only did we look at CPT1, we also looked at CACT and found that in subjects with type 2 diabetes, CACT was decreased. So what does this actually mean for the biology of the cell? If we look at this as a ratio of CACT to CPT1A, we see that this ratio is decreased in type 2 diabetes. And if we just think our way through this pathway, if we have increased CPT1, that means we're getting more and more fatty acyl-carnitine being made in type 2 diabetes. But then CACT is decreased, which means that this extra or excess fatty acyl-carnitine cannot be transported into the mitochondrial matrix in order to be burned as fuel. And therefore, we end up having excess fatty acyl-carnitine. So what we can take away from the slide is that immune cells from subjects with type 2 diabetes produce excess acyl-carnitines. So we really wanted to know if this was true, because this was kind of anecdotal, this is observational. What we wanted to do was take healthy cells from lean patients without type 2 diabetes and see if we could mimic the changes in type 2 diabetes lipid transport within these healthy cells. So as I mentioned, we have high levels of CPT1A, and we would get excess fatty acyl-carnitine. To mimic this, what we wanted to mimic this, we added excess fatty acyl-carnitine. So this we did exogenously into the media. However, with a healthy cell, we would have plenty of CACT to transport it into the mitochondrial matrix to be burned. So to prevent that from happening, instead of having the reduced CACT that we see in type 2 diabetes, we actually used siRNA to knock down CACT. By knocking down CACT, we get this increase in fatty acyl-carnitine that we observed in cells from subjects with type 2 diabetes. So as a reminder, in human type 2 diabetes, we know that there's Th17 type inflammation. They have a cytokine profile that secretes IL-17, IL-21, IL-22. And the big question is, if we force these healthy cells to look like cells from subjects with type 2 diabetes, do we end up making these cells secrete IL-17, the main indicator of Th17 cells? And we analyzed our data by flow cytometry. So here at the top left, you see our control, where we just have our immune cells, flow cytometry dot plot. Each dot represents an individual cell that we're running from these healthy patients. On the x-axis, we have IL-17A, and on the y-axis, IL-17F, both a type of cytokine from Th17 cells. When we treat the cells with acyl-carnitine, we see that there really isn't a difference in the number of cells that are secreting IL-17. If we knock down CACT, that translocase is important for getting the acylcarnitine into the mitochondrial matrix, we also don't see a big difference or any difference in IL-17 production. However, if we combine the two, we knock down CACT and we add exogenous acylcarnitine to mimic the lipid transport we see in type 2 diabetes, we see that there's an increase in the number of cells that are secreting IL-17. When we quantify this, here to the far right, we see that this is statistically significant among several different samples from several patients. So to summarize, we can basically see that if you have excess acylcarnitine, even in a healthy cell, you're able to support increased levels of IL-17 secretion. So just to recap everything I went over, normally with non-diabetic cells or cells from healthy patients, there's metabolic homeostasis. But what we found in immune cells from subjects with type 2 diabetes is that there is normally increased level of free fatty acids, usually due to diet. Once inside the cell, these free fatty acids interact with elevated levels of CPT1. So we get a lot of excess acylcarnitine made. This excess acylcarnitine, instead of being efficiently used and burned as fuel via fatty acid oxidation, is shunted out into the cytoplasm because of decreased levels of CACT. So thereby we have disrupted lipid flux. And what we believe in the future that we want to work on is kind of picking apart the mechanism by which this excess long chain acylcarnitines are able to drive Th17 cytokine secretion as we're able to see. So overall, we know that fatty acids, not glucose, not branched chain amino acids, are driving inflammation in human type 2 diabetes. This has a great implication for interventions in type 2 diabetes management in the clinic. Currently, what the gold standard to do is to make sure that we're managing blood glucose over a long period of time. But this data and our results could potentially explain why we have people who manage their glucose relatively well, but then still have progression of their diabetes down the road and over time. So it'll be really important in the future to consider managing not only the glucose, but also the lipid profiles that we see within subjects with type 2 diabetes. And additionally, when trying to develop strategies that target inflammation in type 2 diabetes, to focus it more on Th17 inflammation, focusing on multiple cytokines rather than just one, and then also maybe using some in vitro assays to test a different type of inflammation as opposed to what we see in mouse models. And lastly, just updating our mouse model for type 2 diabetes analysis. It would be great if we could see if we could change our mouse model to more so mimic what we see in humans in order for it to be more effective. So with that, I'd like to thank my current PIs, Mark Lawson and Pamela Mellon, who have been very supportive here at UCSD and basically helped launch my career in academia and research. I also want to thank them for giving me the freedom to actually finish this project while I was here at UCSD and after I had finished my postdoc at Boston University. I also like to thank my past PI, Barbara Nikolajczyk, and everyone who contributed to this work. And I'll take any questions now.
Great talk, amazing, and congratulations on starting your new lab.